The human genome contains around 20,000 protein-coding genes, many of which have not been associated with a specific function. CRISPR-Cas9-based whole genome screens are a versatile tool to study the function of virtually all genes. To this end, one gene per cell is knocked out in a large pool of cells, ultimately interrogating the whole genome, and the effect on a specific phenotype can be measured. For example, certain knockouts might affect cellular fitness. However, analyzing the function of essential genes is experimentally challenging because cells will quickly die after gene knockout. To be able to also study essential genes, we developed a screening assay where Cas9 expression can be tightly controlled, enabling us to study immediate effects of each gene knockout. We use this approach to study how MYC, an essential transcription factor and master regulator of cell growth, is controlled in the cell. To this end, we combined our inducible Cas9 system with an intracellular staining for endogenous MYC. Surprisingly, one of the strongest hits in our screen was a small and poorly characterized protein called AKIRIN2. Knockout of AKIRIN2 led to a drastic increase in nuclear MYC protein. While mRNA levels were largely unaffected, MYC half-life was strongly increased, suggesting that AKIRIN2 is involved in protein degradation. Indeed, using mass spec, we found that not only MYC, but hundreds of additional proteins were stabilized. To our surprise, all of these proteins turned out to localize to the nucleus, suggesting to us that AKIRIN2 regulates nuclear protein turnover. But how is AKIRIN2 involved in protein degradation? To understand this, we pulled on AKIRIN2 and identified interacting proteins via mass spectrometry. Strikingly, we found that the entire proteasome associated with AKIRIN2, and together with the Hasselbach lab at the IMP, solved the structure of the AKIRIN2 proteasome complex using cryo-EM. This suggested that AKIRIN2 was a direct regulator of the proteasome. But why are only nuclear proteins stabilized after loss of AKIRIN2? Proteasome activity is required in both the cytoplasm and the nucleus, so we hypothesized that AKIRIN2 might be required for translocating this large complex into the nucleus. To test this idea, we used confocal microscopy to analyze proteasome localization. And indeed, AKIRIN2 knockout cells were entirely devoid of nuclear proteasomes, demonstrating that it is required for proteasome import into the nucleus. This in turn explains the accumulation of MYC and other nuclear proteins in the absence of AKIRIN2, which ultimately leads to cell death. These results highlight the power of our screening approach, and we hope that using this system we can further expand our knowledge on the processes most essential to life. If you're curious, check out the paper.